Okay, great. So I've put together a bit of a presentation of what I know about what's being done in genomic surveillance at the moment. So hopefully it'll be helpful in planning the project moving forward. There we go. So first I wanted to talk a little bit about the global sequencing effort to see where we are at this stage and the kind of information that's out there. This is a view taken from MicroReact, which shows all the countries in the world that we have COVID genomes from already in the top left. And then the top right is a phylogenetic tree showing how they're all related. At this stage, the tree is getting quite crowded. So unless you've got a specific question in mind, this can be a bit overwhelming. And also in the bottom here, we've got a timeline showing sample collection over time, which is completely flooded with samples now, which is positive, I think. So there are now about 160,000 publicly available COVID genomes worldwide, 8 of which are from the Philippines. So I'd start off with sequencing in New Zealand. This is kind of an interesting case because we've had a little over 2,000 COVID cases total. There was no community transmission. I'm not sure if that's still the case, but yeah, they're in a pretty good situation here. So because cases are so low, the aim in New Zealand is to sequence all positive cases and to get a really detailed understanding of how the infection is spreading. Currently, new cases are rare, and if one happens, it's sequenced ASAP and compared to all other genomic data that we have, both national and global, to try and understand where the cases come from. And people are using the sequencing to ask questions like, where in the world might a new case have come from, given that at the moment they're probably all international introductions. Can a specific person be identified as the origin of a new introduction or outbreak? Can we use genomes to understand if a spike in cases has been caused by sort of a burning ember or a person that we hadn't detected as being infected with COVID, but who was a driver of transmission? And if we see a spike in cases in the country, are we dealing with a single introduction to the country, or are we looking at multiple introductions at once, which might suggest we need to rethink our border control policies? So this is something that's come out recently from the Lancet, sort of giving an overview of COVID in New Zealand. It really focuses on sort of early March to late April, because that was when most of the activity was going on. So on the left hand, you can see sort of the spike in cases that happened, broken down by imported, import-related, and locally acquired. So even at the peak, most of our cases were related to import from overseas. And on the right, you can see a breakdown of basically all the outbreaks that we had, which is really nice to see on one page, which were mostly well contained, but were still a really big deal, and the demographic breakdown of those. So this is an example of some of the work that's being done using genomics. This is a particular outbreak that made the news. Everyone was very interested. It's called the Auckland Cluster because it happened in the city of Auckland. So you can see there are kind of 3 major centres where samples were collected in the map. And then we've got a phylogenetic tree of our samples in the middle here. And one thing that's really worth noting here is that, you know, these are a lot of cases. We're very interested in understanding what's happened. But if you look at the viral genome, cases from different people are often identical in sequence. So you've got limited ability to really draw together a transmission chain, given the relatively slow mutation rate of the virus. Fortunately, because we had relatively few cases, we were able to really dig into the epidemiological data. So cases were broken down by households and workplaces. Additional data was gathered to figure out if there were any contact links between these people. So here you can see a nice network diagram where bubbles are coloured by the different genome sequences that were identified in different places. And then things are drawn together with links where we know these people had contact. So you can see a slightly clearer picture of how this infection might have spread, which gives you a better idea of which situations we should be looking out for that might increase the risk of transmission. This is another really interesting one that's just come out, which having just flown across the world to get here was a bit concerning. So this is an example of a series of cases that were identified in a managed isolation hotel. So these people did not escape out into the broader country. But what it looks like is that people from around the world got on a flight at Dubai to go to New Zealand. It's an 18-hour flight. Two people who were sitting close to each other had boarded from the same country and were infected with COVID before they got on the plane. It looks like 4 additional people were infected who were sitting near them in the plane during the flight. And then one person was subsequently infected because they were a close contact of someone else in the isolation hotel. So again, in the top left, you can see the phylogenetic tree isn't giving much away. Most of the sequences are again identical. So here, the seating plan was really important, knowing which countries people had originated from and when they had close contact and when they didn't, and also monitoring the onset of symptoms and regular testing once people had entered the country. Kind of the only way that we would have caught this and been so sure about what had happened. Another thing that I think was really cool about the New Zealand response was some really enthusiastic science communication. So some of the key microbiologists in the country, in particular Susie Wiles, teamed up with media to create really digestible explanations of genomics and of viral transmission to try and get the public on board for why we're doing all this testing and why we're doing all this sequencing. And they produced some really nice sort of news media articles to go along with news stories to help the public understand what's going on. Okay, so moving on to the UK, now we're looking at a country that has about 1.5 million COVID cases. And while the UK is very well resourced, it's got great sequencing facilities. Initially, I think we had wanted to sequence every case, but as time went on, we realized that that was not going to happen. So the goal sort of shifted toward sequencing enough samples from enough people to get a representative picture of what's been going on in the UK. So saying that, at the moment, about 110,000 of the 160,000 global genomes have been sequenced in the UK. So this is a very impressive effort, but it's still just kind of a snapshot of what's been going on. This has really relied on COG UK, which is a network of sequencing centres all over the country. No one place could have done all of this sequencing. So it's a really collaborative effort that involves a lot of regular contact, a lot of development of shared protocols and data collection strategies. So a lot of people just completely dropped their jobs in the UK to join this consortium and put in a lot of work to do this. Sequencing is, they say, close to real time, which means 24 to 48 hours turnaround. And once that data comes in, it's interpreted and used locally to help with infection control, but it's also looked at on a national and a UK level. So the sampling strategy, I've never had a really succinct explanation of what it is, but the general description is that it's designed to enable broad population level analyses, so what's going on across the UK, but also looking at targeted analyses of specific populations and allowing freedom to tackle local priorities. So while we want to sequence everyone evenly, there are some situations that we really need to understand better, like a surprising outbreak, where additional resources may be sort of shuttled into that. So every single week, the data are collated and put into a report, and key figures analyse that report to help them with their decision making. And in that process, they'll be looking at local transmission versus imported cases from other countries and from other cities. Rates of epidemic growth, so are we likely to see more cases in the future, or are we doing a good job? Reconstructing spatial movement of the virus, so not just has it got into the UK from overseas, but if it got into London, where did it spread, versus if it got into a smaller part of the UK with fewer people, does it spread the same way? Looking at chains of transmission and what that tells us about social distancing strategies and other lockdown measures. and also tracking genetic changes in the virus. And what's wonderful about this project is that the sequence data are all uploaded to the ENA and GISAID, so they're all publicly available for other people to compare to. So you can see a map of the UK with all the different sequencing and collection sites. It's very dense. They've done a very good job of achieving that spatial resolution. And you can also see a plot of the frequency of different major lineages of COVID over time, which sort of shows that B1.1 has been generally quite successful over a long time. It's persisted, whereas other ones seem to sort of rise and fall over time. Here's a little bit more about the spatial patterning of different COVID strains. So on the left, we've got sort of broad UK lineages, which are just bits of the tree that seem distinct. And you can see some of them sort of pop up in England and then move to Northern Ireland or Wales or Scotland. So there's a bit of spatial patterning in the different countries. But on the right, you can also see that within that global COVID tree, if you start zooming in, bits and pieces of the tree also seem to be restricted to specific regions, which really shows the importance of that local transmission of person-to-person contact that occurs much more frequently within cities. Some centres in the UK are doing even more dense sequencing than the sort of population level sampling. For example, a hospital in Cambridge did a large amount of sampling. They produced 1,000 genomes from which they got about 750 high quality ones that they could analyse. And this allowed them to look at infections in different wards of the hospital, how they were related, and what that told them about what their infection control measures were and what they should do next. So in the tree, you can see the first bar closest to the tree shows some different colours and then a lot of grey. The different colours are different wards in the hospital, and grey samples are samples from outside the hospital. They may be from the community or they may be from other countries. So you can generally see that samples from the same ward cluster together in the tree, but sometimes they're interrupted by cases outside the hospital. So that might be a case where a strain is coming in and out of the hospital via community contacts, or it might be that something's going on in the hospital and a visitor has picked up an infection during that time. So this is all fed back to the clinical and infection control teams, and it has led to decision making around what to do next. There's also a Norfolk study. So this is just studying a big region of the east of England. There's a very big sequencing centre there, so they were well positioned to do that. A couple of observations they made was there is one mutation which has better support than any of the others I've seen for being important, and that's the D614g mutation. There's some evidence that it increases transmissibility, and in Norfolk they found that by May 95% of samples that they sequenced had this mutation, which does support the theory that this aids transmission, but could also be a founder effect. If whatever entered the UK to begin with had this mutation, that might be an advantage. Another thing they noted was that typically you have sort of A and B lineages of COVID, A being sort of the original out of China strain. There wasn't a lot of that found in Norfolk. I think they only found 2 lineage A samples, which suggests that whatever was getting into that part of England was more likely to be coming from other parts of the UK and from Europe, which isn't too surprising. In hospitals, one way that this is being put to good use is to tell the difference between, if you have a spike of cases in a hospital, is that because of transmission within the hospital, or is it because a lot of different cases are coming in from the community? Clearly that's going to have an impact on the types of interventions that you recommend. And the sequencing actually identified an unexpected outbreak that they wouldn't have thought to look for, and that was one across 6 different long-term care facilities, where they saw a common strain amongst these 6 facilities that wasn't found in the community, suggesting there was transmission going on between the facilities. And again, samples showed a clear spatial structure where you tend to find one strain in a particular region. There was also a review of long-term care facilities that came out recently, because this is obviously a very high-risk environment for people when thinking about getting COVID. What they found is that usually within a facility, staff and residents share the same strain. One thing has gotten in and it's spread quite successfully. Multiple studies identified transmission between different long-term care facilities, which is concerning. And again, concerning, it was generally found that if 2 people in a long-term care facility were showing symptoms of COVID, usually by that point, COVID has already spread quite widely through the facility. So if you're waiting to spot symptoms before you employ infection control, you've left it quite late. Proactive testing is definitely a better route. One lineage usually predominated per facility, and it persisted over time. So whether that means most of the time people are doing well and not allowing introductions, but once one gets in, it's really successful, or whether one tends to be more competitive than the others. I think that's a bit harder to say. And even within a facility, if people are in neighbouring bedrooms, they're more likely to have the same strain than if they're across the hall or in a different unit. And some people have said that they can do temporal analysis of the genomic data to figure out when the strain was introduced. Bearing in mind that a lot of these genomes are identical, there's limited temporal signal in that data, but it's probably still helpful. And a big one for us probably is the authors of the review noted that a lot of the bioinformatic methods were inadequate or misapplied. So they didn't feel that the methods were producing trustworthy data that you could use to draw the conclusions that were being drawn. So their big no-nos they identified were assembling amplicons, using poor quality sequencing data to do the phylogenetic analysis, and imputing reference bases where bases were missing. So if you didn't know what it was, you just assumed it was the reference. It can be hard to know what the consequences of this would be. Here's a nice figure from a Washington study at a long-term care facility. This is a floor plan of the facility, and they found a number of different sequences, which they've each given a letter. And here you can see in the floor plan, individuals who are close to each other tend to share a letter more often than individuals who are in different parts of this facility. Mostly a lot of B's clustering together and a lot of H's clustering together. So moving on to what's going on in the Philippines. Last time I checked, in the publicly available genome data, there were 8 samples, all collected around sort of March, April. I know there have been more genomes sequenced, but I don't think they quite made it to the public repositories. So if you look at the timeline on the bottom, across the full time that we could have been sequencing COVID, these were in a very narrow time window, right around that peak. And if you look in the tree at the brown dots, you can see they're all in quite a similar part of that tree, so they are related to each other. But if you take a closer look at that bit of the tree, we can see our brown dots from the Philippines are separated by a lot of other points in the tree, which are samples from other countries, suggesting that what's being sequenced here is probably not a single introduction to the country. It's multiple independent introductions of related strains. So that's kind of some applications of things that people have done. I want to talk a little bit about the bioinformatic tools that people are using as well, with a key caveat that I have not worked a lot with Nanopore Data. So producing really nice polished assemblies and SNP calls currently is not my area of expertise. I can ask other people who are really good at that, but I'm not going to provide strong opinions on that because I don't have any. So the area that I tend to work on is you've got a genome. What do you do with it then? How do you interpret the information that you have? So the first tool that seems to be quite popular and is undergoing active development is called Pangolin. This is available through a website, and you just drag and drop your genome into the browser. And what this tool will do is tell you what global lineage your isolate is from. So once you've dropped it in, it'll say it's a B1.1 or whatever, and then it lets you look at a microreact view of B1.1 to see where it is in the world and how long it's been around for. But it won't place your isolate into that picture, so you have a limited insight into how your isolate relates to others around the world. Kind of the next step up in terms of sophistication is called Llama. This is software that you download instead that does a few extra nice analysis steps. So for this, you also need to download a global tree and an alignment of all the global samples and some metadata as well. But once you have that, you can then provide your own samples, and this tool will sort of insert your samples into the global tree and give you a more detailed picture of how they're related. So this is some of the output from the report. I just looked at their example report here. So you get little mini trees of where isolates sit. You don't get that big overwhelming global tree. So at the top you can see one of the mini trees that shows the context around some of your samples. The one problem I have with this tool is that if there are a lot of identical genomes from elsewhere in the world, it'll collapse them. So it'll say there are 43 samples with this genotype, but it won't tell you where they're from or when they were collected. So some of that insight, I think, is lost there. You can go back and dig that information up from the data, but it's not in the report. And another really nice thing it does is show you mutations that have accumulated in your samples relative to the reference genome. So if you're interested in any kind of genetic markers that will identify this introduction, or if you're looking for specific mutations that are showing up in the literature that everyone's interested in, this can help you see those. So that's kind of the, I would say, a mainly UK developed project. The sort of American equivalent is Nextclade. It can be a browser tool, or you can download it and run it locally. But basically you take your genome that you've got, you can drag it, drop it into your browser, which is quite fun and easy. And again, it'll tell you what major lineage your sample is from. It will place your isolate in the global tree, and it lets you look at mutations that have accumulated. So here is an example where I just took 3 genomes, one of which is the reference, and threw them in. So up the top, we can see some basic feedback on what we've given. We know the sequence names that we've put in. There's some QC that's run. I got these from GenBank, I think, so the QC is quite good. We may not expect to see this all the time, probably not. You get to know which major clade your viruses are from, and stats on mutations that have accumulated. And again, you can see across the genome of the virus where different mutations are. And then on the bottom here, you can see the global tree view with our 3 new samples added in. My one gripe with this tool is that I could only see 2 of my samples. I don't know where the other one is. And the visualization isn't very interactive. So you can sort of zoom in on a part of the tree, but you can't sort of manipulate it to try and find your 3rd sample. So it's not my favorite, but I think there's a lot of really good stuff here which would really help you interpret your data. There's also Civet. I have not tested this because it is quite a complex pipeline with a lot of file inputs, but this is sort of the next step along in terms of if you were to implement this in a public health setting. It's really helpful in the long run to have a pipeline that takes in data every week and produces an interpretable report at the end. So a lot of these workflows are popping up, built around sort of collaborations between people who do genomics a lot and people who need to make important decisions, to sort of take the raw data and remove a person who has to painstakingly manipulate that and put it into a human readable format. It's just made routine, and it's made very clearly comparable between different time points. So that's one example of that. And finally, there's microreact. So this is featured a little bit in the previous slides. It's It's used a lot in COG UK, so sort of Llama and Pangolin also sort of link in with microreact and let you play around with the data a bit. But this is also a really flexible tool. So it's a browser tool. You can drag and drop your files into it. And what it typically takes is a phylogenetic tree and a metadata file. And I find it works best if you have the tree, latitude and longitude of where the samples were collected, and the date of collection. So you can get your map, your tree, and your timeline views. And this is where you really start to be able to do some sort of high quality epidemiological analyses. And what I really like about this is it's extremely interactive. You can view subtrees, you can change the tree around, zoom in, zoom out, just look at a time window, play through time. Lots of things like that mean if you're interested in a sample, you can view it in a lot of different ways, and you can produce bespoke visualizations to really help you communicate your point, and you can share them with a URL. So in terms of really understanding what's going on with your samples, I think this is quite a helpful one. Finally, I thought I'd talk a little bit about the quality of the analysis. So a lot of people are sequencing genomes. The value that you can get out of those genomes really varies. So one of the key things that keeps coming up is make sure you can trust your genomic data. So using recommended software, using recommended pipelines to make sure, particularly if you're doing a phylogenetic analysis, that you can trust whatever tree comes out of that. And also, as you saw a bit with the New Zealand data, because the virus actually mutates relatively slowly, there's only so much you can learn about transmission and an outbreak from sequencing the genomes. So having epidemiological data is also really important. You know, where are these cases? When did they occur in time? Do they have any history of actually being in contact with each other? That becomes really important when you want to move from saying you've identified related cases to actually making recommendations about how to prevent similar events happening in the future. And also, of course, the more genomes, the better. And sampling strategy is really helpful as well. So I think there have been a variety of sampling strategies. Some have been really focused, like there's an outbreak, we want to understand what happens, so we tried to sequence everyone involved in it, versus a sort of population study where you try to balance your sampling across everybody who you're interested in. So that's, I guess, something to talk about in terms of where we want to go with this and how many genomes we're sequencing. I think it really critically involves clinicians and hospitals and thinking about what questions actually need to be answered and what you could realistically do once you've got your genomes. So I think that's it. Really happy to take questions and discuss this more. Yeah, it's over to you guys.
Thank you so much, Dr. Wheeler. So we posted some questions in the chat. Some of them are sort of similar.
Okay. Okay, how to deal with gaps in whole genome sequencing from Nanopore data. I'm probably not the best person to answer this. I posted a link to a Nanopore users conference that's coming up really soon. I know there are a lot of enthusiasts there who are very good at dealing with this. I guess from our point of view, gaps are problematic, as you saw in one of the critiques. If there's information missing, filling it in with the reference is frowned upon. So I think if we don't have good coverage, it is going to be harder to draw conclusions about what that data means. For hospital transmission, how many samples and what course of period would be ideal for sampling to capture transmission patterns? Another hard question. I think, I don't know, I think with the long-term care facility and the hospital sequencing as well, I mean, ideally you'd want to sequence everyone who you think might be involved. That really depends on the scale of your outbreak or your cases. within the hospital, and whether you're doing testing of asymptomatic people or whether you're only looking at symptomatic people. To capture long-term transmission patterns, you want your sampling to be as even as possible. I could see some argument being made for looking preferentially at staff as well, to see, you know, these staff are hanging around for a longer period of time, and if someone was going to be sustaining transmission, it's probably someone who's a regular resident of the hospital, whether that's staff or whether it's patients who are under long-term care, although staff tend to be more mobile. And yeah, llama, etc. Do they require whole genome sequencing, or can you use single gene data? I think with the mutation rate of the virus, even the whole genome doesn't give us a lot of resolution, unfortunately. So if you're looking at a single gene, it's going to be tough. You're probably going to find a lot of identical genes in those 160,000 genomes. So yes, I'd be very cautious. How do you prevent bioinformatics methods mistakes? Everybody makes some, I think. At the moment, I think the community is really trying to be very open and discussing this a lot, because nobody wants to see bad genomes making it into analyses. And I think people are kind of getting a bit frustrated if they see a suboptimal analysis showing up on bioRxiv. So I think there are a lot of sort of recommended pipelines that have been developed and vetted. And I think you're very wise to stick to those, especially if it's a bigger figure in the field who people would be more reluctant to question, but who do get more active feedback about problematic issues when it comes to dealing with COVID genomes. So dealing with someone who's been doing a lot of this and who's been getting a lot of feedback and tweaking their methods is probably going to save you a lot of time and a lot of grief. Again, what's the minimum number of genomes to be useful? I think it depends what you mean by useful. There is, I think, okay, I don't think I need my slides anymore, so I'll stop sharing so I can see your, there we go. Okay. I think for a proof of concept, even one genome is great. But in terms of answering questions about transmission, things start to get a bit trickier. For me, you know, I've dug into the UK data a lot, and I've designed public engagement activities around it, but really your ability to make a strong statement about what's going on with that patchy sampling can be limited. Whereas if you're focusing on a single event, it can be that you can do that more thorough sequencing and really understand one thing that's going on. And I think there is a lot of interest in, I think that flight from Dubai to New Zealand is a particularly good one, because we were being told that it was quite safe to fly. Risk of transmission was low. That's probably still true, but it's important information. There are similar studies with long-term care facilities and hospitals where if you've got an enclosed space that you know you're particularly worried about, having that fine-grained information about what's going on is really useful. But I think at this stage, given that we have 8 genomes from the Philippines already and they're in one bit of the tree, even just seeing the rest of that tree being populated later on could be quite interesting, or whether there is sort of one lineage that's stuck around and the others have died out. Yeah, without genome data, we don't know. We also have one, are there studies related to virus strains and transmissibility or symptom manifestation? Yes, there are quite a few, but if you follow bioinformatician chatter, I think people are extremely skeptical of these, particularly if you've got biases in your sampling and the types of people who you are likely to be collecting samples from. So I think there's some very nice Twitter chatter at the moment about recent studies that have come out where experienced bioinformaticians are very frustrated at conclusions being drawn. Yeah, if you've got it, yeah, I think it's very hard unless you've got a really nice sampling strategy that really hasn't been deviated from, to say this particular variant of the virus is related to adverse outcomes independent of where it's found, when it was found, who it's found in. You know, if something shows up in a care home, it might just be more likely to cause mortality because of who it's infecting. And that's something you have to be very careful with.
Any other questions? All right. So anyhow, just
have a
go
question.
ahead. Yep.
Yes, Nicole, because our collaborator, Dr. Kat, wants to look into hospital transmission because she thinks that that kind of study will be more like practical, useful for their hospital. So that's why I asked that question earlier. Would it be possible to include you in the design of the study so that we can properly allocate the number of samples as well as who will be sampled? Yeah, things
Yeah,
like that.
I'd be happy to be involved.
Also, we have just for information, we discussed last time with Khalil and Ineke. We found out that we cannot reuse barcodes if we reuse the flow cells because of the carryover. So unfortunately, Khalil doesn't have a budget yet to buy the 96 barcoding kit. So we are stuck with 12 barcodes per kit for 2 flow cells. So as of the moment, we can only run 24 samples.
Okay, that's really good to know.
Well, you know, it's like there's gonna be like a step-by-step thing. So the pilot, and then we convince them we could do this, and we could raise money from that perhaps. Maybe like you mentioned, there's also local grants. So from a 24, maybe you could increase to the next one. We could buy the 96 barcodes. We could still use the same Minayan flow cells. So yeah, I guess step by step. So, you know, what's the best we could do with the 24? Here's the question right now.
Yeah, I think it's going to depend a lot on what's going on in the hospital, how much COVID there is, where it is, what interventions are available, and how would they be influenced.
A lot of COVID in the hospital.
Oh, no.
Because it's the main hospital that caters to COVID patients in our region.
Yes, just very recently, there has been a spike of cases in Davao City. So maybe that's also interesting to look into. Maybe, I don't know.
Yeah.
Maybe you can look at index cases with no correlation or contacts, just so you have an idea of like how many lineages are currently circulating. For starting for the pilot, I'm not sure. What do you think about that, Nicole?
Yeah, how confident are you guys in your index cases?
We're gonna have to consult with the clinicians on that.
I think identifying an index case is quite an achievement. Especially, I don't know if you guys are doing sort of proactive testing or if you're waiting for people to develop symptoms. And if people are, I guess most people are coming into the hospital already showing pretty severe COVID symptoms.
They do contact tracing. So once you're positive, then your contacts will be monitored. And I think all the close contacts will be tested.
Okay,
whether
cool.
whether you have symptoms or not. Yeah, so the close contacts, the next level, they will all be tested. That's, I think, for sure. But the rest, we just wait until the symptoms come out.
Just a thought. Maybe we could like because it was well reported that because the first case in Mindanao was from, you remember Ma'am Lar, the one who came from Dubai, was it? She had like a, yeah, like a very long
Of the
flight.
airplane.
Yes, if we have the sample, I don't know.
No, they don't have the samples anymore.
That's too bad.
We don't buy a bank. They don't buy a bank, so that's the problem.
Yeah, because I was thinking, yeah, we could maybe trace cases from that very original case, or but yeah, but I think also it was reported in the news that there were very many waves of introductions of the virus aside from that, so maybe that would
Yeah, yeah. But
complicate.
remember, Dr. Kat, her interest is really in looking at the hospital transmission.
Okay.
Less of the public, like the population types of studies. Um, the question they want to ask in the hospital is, where do these infections come from? Is it just circulating in the hospital, or like what you showed earlier, Nicole, or is it coming from the community? So that they can, prepare well better for further future infections in the hospital, to prevent future infections in the hospital.
Yeah, I think that's a really good sort of test case if we are thinking of a smaller number of genomes. Hopefully, I'll dig into some of those long-term care facility and hospital studies a bit deeper to see whether I can get anything more specific about their sampling strategy. I can also ask around to see whether the bioinformatics community having strong opinions about that.
I think based on our initial discussion with Dr. Kat, I think what she had in mind was to sequence patients in a ward, as well as staff who tested positive, as well as relatives, including asymptomatic cases. So, yeah, that's the initial plan. Well, we could, for example, with 24, with a 24 limit, we could focus on one case. And then branching out to the contacts of that case. And I don't know, Nicole, help us out.
Yeah, I think it would be really useful to get more information about sort of in a given time period, how many cases are we talking about? And if we can have sort of a bubble of like the core staff, the patients, the contacts, just to have an understanding of, in a perfect world, how many genomes we would sequence, and in reality, how many we are sequencing, to get an expectation of what we think the coverage is going to be.
Okay. Any other questions before we close this meeting, this talk? I guess the next step would be to, Dr. Mura and Dr. Wheeler could discuss, maybe with Dr. Kat as well, to do the
we should
design.
invite Dr. Kat,
Yeah,
so that we could design the study.
Yeah. So we could, as we mentioned before, we could draft first maybe the outline of this proposal. The rough outline, we could share it among us, and then we could have a meeting to discuss that. Cool. All right. So
That's good.
cool. Any last words before we end this?
Thank you for the talk, Nicole. That was really interesting.
Yes.
I want to try those online databases and those resources.
Yeah, it's quite fun just having a play with some toy genomes.
All right. So thank you so much, Nicole. And yeah, looking forward to collaborating with you in this project. All right. Goodbye, everyone.
Thank you for organizing, Khalil.
Yeah, thank you.
And thank you, Nicole, for your time and sharing your expertise.
Oh, that's all right. It's cool to be involved.
All right. Bye.
All right. Thank you.
Bye. Thanks.